Good morning, good afternoon, everyone. It is my pleasure to introduce today's speaker, Professor Eline Slagboom, someone who not only brings immense expertise to the field of molecular epidemiology, but also happens to be a dear friend and a long-time collaborator. Eline is currently a professor at the Leiden University Medical Center, where she heads the Department of Biomedical Data Sciences. She chairs the Dutch Society for Research on Aging. She is a medical Delta professor at TU Delft and a member of the prestigious Royal Netherlands Academy of Arts and Sciences. And as if it was not impressive enough, she is also a fellow at the Max Planck Institute for Biology of Aging in Cologne, and recently joined the Academy of Health and Lifespan Research in the US. Now, what about her research? Her work has consistently been at the cutting edge of aging research, particularly while she started in the areas of genome stability, and she has continued the aging work, and now she's truly a driver of molecular biomarkers of biological age and immune metabolic responses to lifestyle interventions. And the Leiden longevity study that many of you know, the Growing All Together study, and her leadership in the Voila consortium are just a few examples of how her research is shaping our understanding of healthy age. On a more personal note, I have always admired how Eline's passion for science is only matched by her generosity and collaborative spirit. It has been a privilege to work alongside her, and I am excited for all of us to hear about her latest insights today. So without further ado, please join me in welcoming Professor Eline Slagboom. And before I disappear from your cameras, remember that you should post your questions in the Q&A. Eline, the floor is yours. Thank you.
Thank you very much, Jose, for these kind words of my good friend, Jose. And, you know, while we are aging, we have been working in aging research for a long time. And today, it's really nice that you invited me to talk about our work. And I will be talking about the genetics biomarkers and the impact of lifestyle changes in later life. And all my research is very much focused on, in the first place, the human biology of aging, and in the second place, molecular profiles of individuals. So here you see my favorite study member in the Leiden longevity study. She was here 100 years of age celebrating. She was a member of a monozygous twin, and she became 103 and her sister 106. And when I met them, they were, I think, 95. And when we looked into their lipid profiles, they looked like, I would not say young girls, but at least like 30 years younger than they were. And that is really intriguing. So the studies in which I am working to investigate molecular profiles of aging from two sides, the unhealthy path and also the healthy path, I do in the first place in long-living families. So I'm interested in the young metabolic signatures that they have and the low-level inflammation, and I'm really eager to find out what kind of key processes drive their healthy aging patterns. Because, of course, you know that we all became to live very much longer than 200 years ago, but it also came with a price, because with our long life come the increased risk for all of these diseases that you see here. And so the real research is not about how to get people from the 100 to the 120. The real research is how to get as many people as possible in a healthy way from the 60 to the 80. Because many people do not have such healthy aging profiles as as my favorite Leiden longevity study members. There's also a lot of clinical questions. I'm working here in the Leiden University Medical Center, and of course, these centers are now being flooded by older patients that have usually a lot of comorbidity, polypharmacy. There's a lot of heterogeneity among them. And so there's a strong sense that we should know more about aging and be able to kind of classify older individuals. So although we live so much longer, by the time that we get 65 and we're thinking about our pension, already more than 60% has two diseases or more. So this multimorbidity is, of course, a major problem in getting healthy from the 60 to the 90. And this percentage is even higher in the US at the moment. So what is our aging research about then? It's a kind of a triangle of interrelated research. So on one hand, we try to understand mechanisms of aging and longevity, of all the protective mechanisms, but also of the regular aging process. We also want to mark vulnerability and how fast your aging process is going. Because how can we study older individuals if we cannot group them into faster and slower aging people? And so we are making biomarkers to try and group people as they age, but not only to indicate the risk of individuals, but also how they respond to interventions. And there, of course, molecular epidemiology could pay a nice contribution. And the moment that you have the markers that would indicate which people are at risk of faster aging processes, then you want to do something about it. And you do not want everybody to enter the clinic. Of course, the first stage would be whether there's lifestyle interventions that could lower all the risk that these people have. And there's, of course, a big behavior component to that, but also a very molecular scientific component, because people respond so heterogeneously to lifestyle interventions. So let's start first with the top part, which was the more mechanistic insights. So here you see Niels van den Berg, who came into my lab at a moment where I kind of almost gave up to study the genetics of longevity, because we had done worldwide studies of long-lived people, all unrelated, case groups, control groups, investigating this by this genome-wide association studies. And we could find not many more genes than just the ApoE gene, and we knew already about that gene. So I was wondering, where is the heritability of longevity? And then Niels van den Berg started to work with databases based on demography. So all of these people died, so millions of people that died. And he investigated all the time 3 generations and was all the time wondering if people belong to the top survivors in a birth cohort, do they transmit this ability to the next generation? And what he found is that you really have to go to the top 10% survivors of each cohort, of each population. And there you will find heritability. If you go to the top 10% survivors, you can see that also their children and their grandchildren, they become longer survivors. They have a bigger possibility to become long-lived. And in fact, he found that that acted as a quantitative trait. And so with every ancestor that lives into the top 10%, whether it's an aunt or an uncle, even if your parents died younger because of environmental components, then if you have very long-lived aunts and uncles, you will have a higher probability to become long-lived yourself. So there we thought, now we can try to find the people that have the more heritable longevity. And he found that whether you went to Utah or to Zeeland, all demographic data sets, let's say the Utah population was much more healthier than the Zeeland population. The answers were the same. You have to go to the top 10% survivors of a birth cohort. So then Niels made a score. He called this the longevity relatives count score. And I know also other scores have been made, but he made it like this. For every individual, you count the number of ancestors that you know when they died. And then it's a weighted number of the top 10% ancestors. So those that live into the 10% longest lived of their birth cohort. divided by the total number of ancestors. And then he also put weights on this, and the weights are depending on the genetic distance. And so whether you're talking about parents, aunts and uncles, or even further relatives. And so, for example, this LRC score of 0 means that you have no long-lived ancestors. If you have a longevity relatives count score of 0.5, it means that 50% of the ancestors that you could get a hold on the data are long-lived, live into the top 10%. Okay, so now this is the last complex slide, and then we go to something more easy. Because the next question was, if you have an increasing number of long-lived ancestors, can you see that the probability to live long yourself increases as well? And this figure is about, on the y-axis, the standard mortality ratio. If it is 1, it means that individuals die at an average of their birth cohort. If the standard mortality ratio is below 1, it means that there's an excess survival, so that these individuals live longer than their birth cohort. And he investigated this in 32,000 individuals. And he observed the following, that when individuals have, and this you can see on the x-axis, which groups of longevity relatives count score he took, when you have when you have an LRC score of 0.3, meaning that 30% of your ancestors are long-lived or above, then you can really experience the higher probability as an individual to also become longer-lived. And this even mounted to a very high excess survival for the individuals with these higher numbers of ancestors that live into the top 10%. Okay, so what does this mean for living individuals? And so now we go to a cohort study of living individuals, the Leiden longevity study. We collected these people already more than 20 years ago, and we included long-lived individuals, they were above 90, with brothers and sisters that were also above 90. So to enrich for, let's say, familial longevity. And then we also collected their children. They were at that point in time about 60 years, and as controls, the partners of the children. And what Niels then did is that he also collected all the individuals in 2 generations that had died. So we know exactly for these generation 1 and 2, when all the brothers and sisters of the sibling pairs died, when their parents died, but also of the parents of the controls. And that was really indicative. Because what we see now is we wanted to know, is the offspring of long-lived individuals living longer than their partners? This was the case. Then we wanted to know, is also the health span longer? And so Niels investigated prospectively what was the incidence of the first metabolic disease of these individuals. And here on the left, you can see in the blue if you wish, the survival curve of when people develop their first metabolic disease. And you can see that the offspring, the blue line, develop later their first metabolic disease, and that is in fact on average 6 years later than the partners. But if you know all the ancestral data, then you can say how much later does offspring develop their first metabolic disease, offspring that have at least 30% long-lived ancestors as compared to partners that have no long-lived ancestors. And then you see the figure on the right, and there the difference between the blue and the green line is much larger. And so offspring of long-lived individuals with 30% long-living ancestors develop 13 years later than their partners their first metabolic disease. And this is about the same kind of gap as if they were the highest and the lowest socioeconomic status individuals in a population. This 13 years difference with your partner is really enormous. This also means that if you try to study the genes that drive these effects, is that what you're essentially studying is the genes that delay multimorbidity. It's not only the first metabolic disease, it was also the second, and it was also cancer. And Niels then investigated also the Swedish registry data for exactly the same question. And there we saw exactly the same. So when you're looking at how long-lived your ancestors became, and when you really focus on the top 10% survivors, then you come to the real kind of heritable cases of longevity. So this we applied on the Leiden longevity study because we have sibling pairs here that we used to do linkage analysis. But now we said we will only do linkage analysis on those individuals, the sibling pairs that belong to the top 10% survivors of their own generation. And that means that they had to be, well, at least above 95 years. And we also include the rule that at least one of the parents should belong to the top 10% survivors. This meant that we could do this linkage analysis in only half of the study. And so although we selected for sibling pairs, half of the studies really did not have these long-lived ancestors. But even if we did on 212 of these siblings, you see here now what was the mean age at death, so they were almost centenarian. Now we suddenly found positive linkage signals. So what these signals mean, I'm not going to explain linkage too extensively, but if there are these 4 regions, it means that in this region under the linkage peak, it's 1,000 times more likely that the longevity gene in this set of people, it's 1,000 times more likely that the gene is there than it is anywhere else in the genome. So we have here 4 locations, and in total there are about 373 genes there, where it is 1,000 times more likely that there is a longevity gene than anywhere else in the genome. Let me just get this little sip. We sequence these families, and at the moment we have a whole pipeline to pick out the most interesting, let's say, protein altering mutations in the genes under these linkage peaks, actually under the 1 lot drop, which are these pink regions. And we are functionally following up these variants. And the first one that is also already very interesting is the CGAS gene that is under one of the linkage peaks. And that gene is very interesting. So also to give you a bit of a glimpse why this could be interesting, it is a gene that is sensing whether the cell is exposed to strange DNA that is not coming from your own body. And so this happens, for example, at infection. A double-strand DNA, then the CGAS gene is increased in expression. So what do you think that these long-lived people carried in this CGAS gene? They carry a mutation. So the mutation is making, let's say, a lower amount of the CGAS protein. And it turns out that that is exactly, it's functional, and that is exactly what these people have. They have a kind of dampened or lower expression of the CGAS gene, also when their cells are confronted with strange DNA. And so let's say the protective effect of the CGAS in this case is a lower expression than in the rest of us. So this is learning us, of course, that a too vigorous response to infection is usually not the best response. That is what the indication might be. But we are still doing the functional analysis in cells and then also in killifish of these variants. So, in conclusion, familial longevity is marked by belonging to 10% top survivors and behaves then as an additive quantitative trait. And at the moment we are including novel families that live up to this rule. And by using the scores, we could show that you increase the contrast between people from these longevity families and other people, controls, so that you're basically looking into the genetics of multimorbidity. And for the group of families that we looked into, we have now 4 regions where it is very likely that longevity genes are there. And we are in total investigating 16 genes at the moment where long-lived people and their family members that are long-lived all carry these variants. So that is where our genetic research is at the moment. Now I want to switch gears because when we go to the rest of the population, what are now the interesting phenotypes to study with aging? And you could say we study calendar age. You would like to study biological age, because if we knew exactly how fast people are aging, then we have a phenotype. And disease, you always have the diagnosis as a phenotype, but here in aging, it's quite a problem. So therefore, many people are investigating different definitions of biological age. And so you could say, if your calendar age at a certain moment of time is the distance between that age and your birth, then your biological age is then, it depends a bit on what you define. You could say it's the distance from the moment where you live till death, or maybe to your first disease, or maybe to some aspects of frailty or multimorbidity. But you always want the biological age to indicate a risk that you will have in the future. And that risk is indicating your physiological vulnerability, right? And at this moment, my lab is very busy in trying to make these markers out of metabolomics measures. Now, this whole principle was already thought out by Alex Comfort a long time ago, in 1969 already. And what he did was interesting, because he took all the different biomarkers that were known in the clinic, indicative of organ system health, and he made interesting, let's say, combined scores out of that. And that makes sense, because your aging rate in different organs will be different. So if you want any kind of readout of the physiological state of individuals, and let's say how fast they go down on the stairs that you see there at the top, then it would be logical to take the best markers out of a combination of tissues. And since then, because so many things change with age, people have made many different kind of estimators of biological aging rate. Some are made at the end of lives, such as the frailty scores. That's more an accumulation of deficits that you have at that age. But also others made scores very early in life, out of the idea that if you want to do intervention studies and you want to see that somebody is aging more slowly, you're probably going to start that earlier in life and not when people are 80. So my group is trying to make these kind of multivariate biomarkers out of molecular data. And so there are two principles in which you can, or a couple of principles, but we apply two principles to make these kind of markers. You can say, I measure my molecules in a whole group of individuals, and out of those data, I'm going to make my molecular predictor of the age of an individual. And I compare that to the chronological age. And so here you would see the principle. Let me use my mouse. You could see here that if somebody is 70 years of age, then on average, people will have a level of the biomarker that you generated that is here in the black point. But suppose that if you measure the person, they have this green point or the red point. If they have the red point, it means that they're actually molecularly older than they are. And if you find the green points, they're younger than they are. So it's an estimation of biological age, you might say. And of course, you probably know this because this was first applied by Hanem and Horvath using DNA methylation. Why? Because there was such a relation of DNA methylation with aging. So they made molecular clocks out of this. And the molecular clocks made out of DNA methylation, they're very precise in predicting the calendar age. And when they then investigated if you are older than, if your molecular clock is older than your calendar age, whether that associates or predicts events for the future, then they saw that there are a lot of different predictions. It predicts increased mortality risk, cancer risk, etc. So My group started this on metabolomics because we felt that we understand the metabolome a bit better than the epigenome. And we also wanted to make these kind of markers out of metabolomics data that many people generate because it's so affordable. Because if ever you want to use this in the population or in the clinic, these cannot be very expensive markers. And we used a platform of NMR-based quantitation, and that's a Nightingale platform, and they generate 226 metabolomic biomarkers, but many have a strong correlation. So in a sense, we're usually working with about 65 of these. And you might say, you know, is that going to be so valuable? Because it may be cheap, you know, very Dutch approach. It may be cheap, but can that be so indicative? And we thought, well, why don't we start here? And then you can always make these kind of scores better. And now years later, there's Daniele Bizzarri. He has made a shiny app where you can, out of this kind of data, calculate for your study the different scores that we made out of this metabolomics data. So the first was Erik van de Akker. We had 25,000 samples measured by this metabolomics data in a very nice distribution over age. So let's say between 30 and 90 years of age. And Erik made scores out of that. And here you can see that, let's say you see on the y-axis the predicted age of individuals, and you can see per age, so let's say at 60 years of age, now the range is very wide. So it's clear that by metabolomics, you cannot so precisely predict calendar age as with DNA methylation. But of course, that is not what we're looking for. We're looking for the difference between the predicted molecular age and the real age of individuals. So when Erik made these scores, he now investigated whether this metable age score also predicted endpoints. And then he saw that especially in the cardiovascular field, a higher score means that you're kind of aging faster in molecular sense, is associated with more risk for all these different cardiovascular events, including mortality. And here you see, here you might say that with every year older of this metabolic age score, you have a 2 to 4% increased mortality risk. And that was very much comparable to these first measures of DNA methylation age. And this was independent of a lot of other important variables, BMI, smoking, diabetes, etc. And the, let's say, the molecules that were part of this score are here a number of amino acids, and then you get creatinine, and then you have the lipids, lipid particle sizes, etc. Okay. Next, we wanted to know whether you can make an improved score if you make the score based on mortality. Because yeah, that's such a big event in aging. We're just wondering that if you make the score on mortality, if you get the better score or the worse score. So Joris Delen investigated, together of course with many other labs, 44,000 individuals that were followed for about 16 years over time, in which case there were more than 5,000 deaths. And there also the metabolomics was measured. And then we found that many metabolites actually associate to mortality risk. That's here on the right. So above the red line, you see many metabolites associating mortality risk, and the ones that are circled were the ones that were already found in an earlier study of Fischer. So next, Joris made a score out of this, and then you try to make the maximum prediction of 5 to 10 years mortality out of the minimum number of metabolites. And he found that if you make a score out of 14 metabolites, then you really predict mortality very well. Here you see the ROC curve. The black line is, let's say, the traditional risk factors, if you really know a lot of clinical aspects of individuals, but the red line are these 14 metabolites. So you can see here now that in this score, it was remarkable that you have now VLDL particle sizes And one very important marker is the glycoprotein acetyls, which is an indicator of low-grade inflammation. And so it's obvious that if you make a score based on mortality, even if it's on the same metabolomics platform that you make the score, it chooses other metabolites to predict the endpoint than if you do it based on calendar age. And we saw that here you see the hazards ratio at different age strata, and you can see that the score really indicates the ratio of mortality, well, quite convincingly, I would say, for many different age strata. And of course, at the higher ages, you also have a bit of the smaller numbers. So then we asked ourselves the next question. So can we show in epidemiological studies what this score represents? Is that just mortality? And because who wants to know just mortality? And also, why does it predict mortality? If it's only these 14 metabolites, how can it predict mortality so well? And oh, I see that some text dropped out, but look please on the right side. So the question is also, can we use this kind of score? Is it actionable? And can we maybe improve the scores? So we first investigated a whole number of scores in their relation to, for example, frailty. So here you see the study of Lieke Kuiper. This was done in the Rotterdam study. It is about 750 people. And you can see here on the left that she tested different predictors. So first you get the Horvath and Hannum clocks. So these are really the first generation methylation clocks. And then you get a number of a bit more interesting methylation clocks that are all built a bit more on on the endpoints, on let's say a bit more in the direction of the phenotypes of older age. And then you see metabolic health here at the bottom and metabolic age. And what you can see is 4 different representations of frailty, so 4 different frailty measures. And you can clearly see that if you take out of the methylation clocks or out of the metabolomics clocks, if you take those markers that were built on physiological endpoints, so either mortality or mortality-related phenotypes, there you get actually the best prediction of frailty. And metabolic health, well, we felt very reassured that even this rather simple measurement was predicting frailty so well. And so then we tested a whole lot of other things. Let me show you a couple of things. So, even kijken of dit. Oh, sorry. So next we tested the, let's say in the PROSPER study, what is the 3 years prospective loss of cognition by all types of different tests. And this was in a study of 5,000 individuals. And later we tested even 10 years of follow-up in a larger study. And the story was all the time the same. If you have a higher metabolic health score, it's a bit counterintuitive, but it means that you have a higher mortality risk. Then also you have a higher risk of cognitive function loss. So it's frailty, it's a loss of cognitive function. And then in the UK Biobank, we also investigated what is the relation to incident pneumonia. So in UK Biobank, the Nightingale data was measured in over 50,000 individuals, and there were 1,250 incidents of pneumonia and 8 years to follow up. And then here you see the representation of another score. This was made, but the results for the metabolic health score were exactly the same. Is that you see on the right side that if you divide the individuals into quintiles, this is of course a very big data set, then you can see that the risk and the prediction of pneumonia risk in the highest quintile of this score is much higher than as compared to the lower quintiles. So it's indicating the risk of pneumonia events. It was even indicating the risk of COVID hospitalization. That was to small numbers at the time in the UK Biobank, but we recently showed that also in other clinical studies. And so you can see that this metabolic health score is kind of an indicator of vulnerability at, I would say, 50 plus years of age. So then the question is why? Why can we predict so well? What is behind it? What is the score picking up? And then we, oh, I do not know how to get rid of this top. No, I just hope that you can read it. In principle, we took 3 different cohort studies where we would have 8,000 individuals in total. And out of each of these cohort studies, we took the top scoring metabolic health people and then the bottom scoring people. And so that is what you see here in the picture, top and bottom for 3 different studies. And then we selected the people based on matching for age. And so now you have top and bottom scoring people that are very different in their score, but the age is the same. So there's not an age difference. And then we asked the question, if you investigate these contrasts by proteomics, can we get a better feel for what the score picks up? Why can it predict mortality so well? And then first we had a Luminex assessment of a number of cytokines that we chose because of other, let's say, publications on the markers of frailty and mortality. And then we saw that in the population, there's a whole lot of markers that you can't even pick up. These cytokines would be better suitable, these undetectable in population-based people, but they're better detectable in clinical studies. But we also clearly saw that the people with the increased metabolic health score, they had also higher levels of IL-6, of MIG, and GDF-15. And GDF-15 is also a very interesting protein because it's associated with many different diseases as well. And then not only the Luminex assessment, but also LCMS-based plasma proteins were clearly associated with this contrast. So an increased metabolic health score is associated with an increase of a lot of proteins that represent inflammatory responses. It may not be surprising, but it's reassuring that what this marker, this simple marker picks up is actually quite a lot out of your inflammatory response. And it also picks up a lot of cholesterol transport issues and the regulation of apoptosis. And so out of all these proteins, there were about 105 different proteins that associated with this contrast. And at this moment, we're investigating in different data sets how the metabolic health now really relates to the proteome data in large cohorts. Well, which of these proteins is predicting the best? Can we add some proteins to the score? Or is it so that if you increase the number of molecules that you measure in blood, could it be that you cannot increase the prediction information because there's a limit to what blood will tell you? So these issues we're investigating at the moment. So now we come to the last part because we also wanted to know, let me have a look at the time. Yeah. So of course you want to know whether this or other scores could also follow the response to interventions. And I was very eager to investigate lifestyle interventions in 60 plus individuals and to approach that in a biological way. So to collect blood and fat and muscle and to do a lot of multi-omics in these people and also a lot of clinical readouts. So this is what we did. The intervention is 12.5% lower calorie intake and 12.5% increased physical activity. And let me get my mouse. Yeah. And we did a lot of measurements also, for example, recording activity data by wearables, blood pressure, DEXA scans, etc., so that we could get an indication of what changed, but also by what heterogeneity in these individuals. And here you can see that in this 13 weeks intervention, you can see that there's significant lowering of BMI in both men and women, in body fat percentage. That was clearly seen on the DEXA scan. And when we look, for example, at blood pressure, you did not see much change in blood pressure, except when people had high blood pressure, which is what you see often, of course, in intervention studies. This study, by the way, is the Growing Altogether study, and it's called, abbreviated as GOTO. So then the question was, and this is work of Fatih Bogaerts, is there an improvement of the metabolic health score by this intervention? And then if you look overall, on average, you do not see anything. It's just like blood pressure. But if you go to the top tertile of the people, top tertile scoring people of the metabolic health score, meaning that is the top tertile of mortality risk, then you see that 44% of this top tertile lowers their risk. So the metabolic health score does indicate a health improvement. A step further is that it is not the most sensitive of scoring of biomarkers. And so out of the same metabolomics, Fatih did other experiments where he investigated if you just take the whole metabolome, then how can you best make the predictors for men and women of this response to the intervention? And then you get different kind of predictors, also showing much more effect than what the metabolic health score is showing. And so I think it's fair to say that we did not really expect that the markers that are best for indicating your risk of becoming vulnerable, that those markers will be immediately the best markers to indicate the response to interventions. That doesn't mean that you cannot use the metabolomics to generate the scores that do tell those things, right? So he made a so-called PLIS score, and you can see here that to make such a score for females to indicate their response to the intervention, you needed many, many more metabolites than you generated this kind of score for men. We see this all the time, is that there are large sex differences. Not so much, interestingly enough, in the response with respect to BMI and fat, but a lot in response with respect to molecular profiles. So in the meantime, we've also investigated methylation profiles and gene expression profiles, and all of these papers are now either being just accepted or being submitted. But I think the last point that is very important is that the PLIS score indicates for many more individuals how they respond to the intervention. So probably if you want to apply this, you need to make combinations of scores. And then there's this last point of, it's not good enough to just look at the diet. It always makes an enormous amount of difference how active people were. And we measured this with the gene active accelerometer. And then Stelios generated again novel scores that would indicate what people actually did, because there were programs available, but with older people 60 plus on a bike, your watch is telling you that you're stacking shelves or something, but it doesn't recognize that you're sitting on a bike. So what we did is that we had older people do a lot of different activities and at the same time record the time for all the equipment that these people were wearing. So all different kinds of wearables. And there were two very nice guys who were liked very well because they were timing all the activities. But of course you saw poor older people doing all these activities and the guys doing basically nothing, but just watching or telling people how fast they should bike or run the stairs or even do vacuum cleaning. But it gave us a lot of data because now we could interpret what people actually did. And this is the last thing that I really want to say about this, because I thought it was already amazing how much people could improve their health only in a 3 months intervention when you're 60 plus. But this is also an interesting one, because here you see the type of activities that men and women did during the day. And I'm not going to explain the whole picture, but you see at the top is the women and at the bottom is the men. And at the top you see a lot of light green and at the bottom you see light blue. Well, light green is a lot of moving around. It's mainly, let's say, doing the household. And the light blue is sitting, being sedentary, reading the newspaper, etc. So you would expect that if the men are so much more sedentary, you know, what was the health gain that they had? But you also see some of the red and some of the dark green being, you know, broader in the men. And it was clear that if the men do their exercise for this 1 hour per day, they're doing it more vigorously. And so here you see where we combined on the y-axis all these indicators of health, and on the x-axis you can see all the different measures of activity. And there's one thing that I want to point out. All the different measures of activity, especially when the ankle wearables were interpreted, were very well associated with all the markers of body fat, BMI, etc. And body composition was then also responsive in both men and women. And so you see that there are these significant blocks in both the left, the female, and at the right, the males. But if we go one block below, there you can see that all the signal seems to be in the men and not in the women. You see in the first place, remarkably, that if you measure only C-reactive protein, that not much is happening. But if you take another marker of inflammation, which is glycoprotein acetylse, then you can see what advantage the men had out of their intervention study is really lowering this low-grade inflammation marker. And it's doing a lot of other interesting stuff in men, HDL, diameter, and so also triglycerides. But it's stunning how little the women had from all these household activities. So this could, of course, have two different possibilities. It's either a physiological difference because men have their muscles responding in a different way to these activities than women. The other possibilities is that women were not doing their exercise as vigorous as men, and that is really a message that you should sort out. We now have the markers that could indicate the difference. So the question is really, you know, what kind of exercise should a woman do to get the same kind of molecular advantage as the men have? And that is now going to be the next studies. So we have a novel intervention in 60 plus individuals that is ready at the end of this year, where we included much more resistance training. So there's also dietary components, but there's much more focus on training in the gym with, of course, guided trainings. And there we can investigate whether that group of women also had more advantage to the intervention as the men do. But I think the most important thing is that we have markers that can indicate the difference. So in conclusion, especially measurable health was a score that is affordable and was indicative of overall health and vulnerability. So you have like all these issues that are mentioned here. So we like to think that this is an indicator of biological age. It represents inflammation, as the proteome studies showed. So we understand now a bit more why the marker is so indicative of unhealthy situations. The lifestyle interventions did change the score, but other markers need to be also necessary in addition. And at the moment, we are investigating whether this can be implemented in population health studies. And so you define the target group at risk because of the measurable health score at the GPs, and then they go into, let's say, lifestyle centers to do the important lifestyle changes. And of course, the future could be that you can do health checks with larger groups of individuals and indicate them to go first to lifestyle centers before you start using medication. We definitely noticed in clinical studies that the measurable health may not be good enough, so we need much more inflammatory-based markers to make distinctions between the different older individuals. And of course, we are trying at the moment to improve our metabolome scores. So with that, I'd like to stop and finish my, my lecture. These are all the individuals that have contributed to our work. So, I don't know, Jose, should I stop sharing?
Yeah, probably yes. Thank you very much for this delightful.
So, oh, I'll put on the lights.
Thank you, Eline, for this wonderful lecture and the presentation of your later results. And now we are opening the floor for the participants, for the attendance, and we have some questions coming. Okay, so we have on Larissa Constantino. Did you also measure muscle mass and muscle mass changes during the with interventions?
Yes, yes. So we did that already in the GoTo study, and in this novel study that is ready at the end of this year, we did it more, yeah, I would say consistently. That study is called the Voila study. But we, what we do see is, and this is of course when you're doing the, let's say the normal GoTo intervention study, there are people that are also losing not only weight but also muscle. So we are quite convinced that if we're doing these intervention studies, that the exercise has to oppose to that, right? So that you really have to do some more physical exercise to not lose muscle. At the same time, even when in the GoTo study people were losing a little muscle, it's not that much, and they were gaining, let's say, inflammatory health, if you wish. So that was also an interesting part.
Thank you. From Daniel from Islo. Terrific talk, Eline. I am curious about the heritability study. It is clear that a stronger signal emerges when you focus on the longest living 10% of ancestors. Did you consider doing a similar study on the shortest lived group to identify deleterious risks?
Yeah, there's a group in that is doing this on the basis of the Swedish registry, because they were interested also in childhood mortality, and but you run into a different kind of question, of course, because many different diseases will shorten your lifespan, and it's quite a different question as the longevity question. What is also an interesting question is, you know, if you know that families have a risk for a certain disease because they have certain mutations, do some branches of families, despite of that, live long? And that's of course also an interesting question where these things come together. But we did not look in this data set for the shorter lived families.
Okay. And continuing with Daniel, for the third study, is it possible that there was a stronger response in males because they were more sedentary than females to start with?
Sorry, is it possible that?
It's possible that there was a stronger response in males because they were more sedentary than females to start with.
Yes, I think that's at least part of it. But then if we investigated the more active males and also the more sedentary females, then we still see these differences. Right? So it was definitely the case that the women were slightly healthier at the beginning, and so that you would have the bigger response in the men. But the sex differences remained in any kind of analysis that we threw to the data.
Thank you. From Shandong Wang, nice talk, thank you. Considering molecular biomarkers, such as methylation of CpG island, or metabolic biomarkers, such as the 14 metabolizing your study in the blood, can often, but may not necessarily be attributed to more aging processes? Are there any molecular biomarkers from other compartments, such as the cerebrospinal fluid, to reflect accelerated aging brain?
Yeah, so you're basically asking how can you even make the better markers when you are not just taking any kind of markers, but markers that are more indicative of certain compartments that you know are aging. And I agree to that. So the spinal fluid is of course not something that we have in these bigger cohorts, but I'm part of an dementia consortium where people have measured a lot of dementia-specific markers, for example, along with the Metabo Health score. And I can still not, I mean, the analysis have not been done yet, but I'm also very eager to find out how different, let's say, more disease-related markers relate to the more aging markers. So what I'm a bit more hesitant about is whether measuring more and more omics levels will increase our predictive information. So for myself, I also expect a bit more from these domain-specific markers, where you, for example, would say, I have specific markers that would indicate mitochondrial function, for example, because that is not very well represented in the metabolomics that we measured. It could also be that if you take metabolon, for example, where you have much more metabolites that indicate the Krebs cycles and all the different true pathways, is that you make the better predictions. But we still have to find that out.
Thank you. From Christopher Whaling. Wonderful talk. The CGAS STING pathway has become a huge area of research in inflammation. But I am curious if there is any evidence for antagonistic pleiotropin, where the better CGAS agers may be more susceptible to infections by DNA viruses.
Yeah, I agree that let's say there's an enormous field. This is why I also mentioned CGAS. I don't know yet. I'm not a CGAS expert, and we're still it's only pretty recent that we really found that the mutation is functional. So in the beginning we did not really take it for granted that what we are looking into that is really so important. But let's say it's whether it's antagonistic or not. I don't know whether it's a good question. I don't know whether we could investigate that in our study, right? So, of course, a mutation that is in a linkage region is present in all the long-lived family members is quite a strong case that this that this is related to the longevity. And if it was, it means that you have an advantage if you have a lower CGAS level, or at least a lower response. So I don't know whether it's antagonistic or not.
Okay, from Arion Meta, what was the magnitude of improvement of Metabo Health score in the top tertile of the GO2 study?
What was the level of improvement? Oh, yes. Yeah, that's even denk hoor. Let me think, because yeah, I can't tell that by heart, because we saw that yeah, I can't tell you by heart. What we're doing at the moment is trying to get all these data together to say, like, in the population-based studies, it is a score that is between, let's say, -2 and +2. And it is scaled at the moment. And what you want, that it gives an individual value. So for that, we need a reference database, which we can make out of the data that are present now in the cohort studies. And we're trying to translate this into, if you have a 1 point increase in metabolic health, what is now the increase in mortality risk, and how much of that value is lowered in 3 months of an intervention study. And we have not calculated these things properly. So it would be too early to say, but this is exactly what we're trying to calculate. And what we're also trying to calculate actually is not only what is your increased mortality risk, but also in the prospective studies, what are you losing in terms of health span? So what is the relation to the first moments of the first disease event, let's say the first metabolic disease, if you have 1 level increase in metabolic health, and then how much decrease, like for example 0.3, do you see in the intervention study? And if you would multiply that by 5 times, would you be able to gain health years if you would do this kind of intervention for 1.5 years, for example? So we're trying to make these calculations, but we were still struggling a bit with the scaling of the score.
Thank you. Well, before we asked Eline if she had to run to her next meeting, she said no. So
No.
there is one more question. And
Yes.
have you examined the relationship between dietary intake and the PLIS score?
Yeah, I think I could not say this because this was the combined intervention. So we do not have, let's say, this separate diet intervention. That is a very interesting question. So what we're doing at the moment is every intervention that has measured a Nightingale data, we are trying to investigate what is the response of the PLIS and the metabolic health and the metabolic age on the intervention. So there's a study called Person study, which was diet intervention only, based on individuals with insulin sensitivity, either by the muscle or the liver. And there we also see that the intervention is especially interesting for the people that have insulin resistance in the muscle when you're looking at the top tertile of the metabolic health score. So we see very interesting things when it was only due to diets and not due to activity. So we try to get a better grip of all these things to know what is the, because I had the impression first that the metabolic health score is very indicative of low-grade inflammation, that it works so well with 60 plus because it indicates sarcopenia, for example, and that is less sensitive to insulin sensitivity. And I believe that's the case. We looked into the Leiden longevity study, and it's, if insulin sensitivity is already becoming a problem before 60, and metabolic health is less sensitive to that. So in my mind, you would need different markers to get a complete picture.
Okay, let me then just finish with 2 quick comments, more than questions. We have been advancing in terms of our knowledge of healthy aging. What ethical changes do you think we might encounter when it comes to access of this knowledge, of this transformation that we are experiencing in terms of equity in healthy aging solutions?
Yeah, and you asked for the ethical, right? Ethical implementation. Yeah. Yeah, I think that's a very important question because what we're doing, of course, is trying to predict everything. To understand it better, because we're making the predictors also to be able to make groups and to understand, well, at least make more homogeneous groups and understand those groups better. But at the same time, if you're making the predictors, then what you're basically doing is telling people you are at risk, and now not for a disease, but for aging faster, for being vulnerable. And you have now a responsibility to do something on your own lifestyle. And if it doesn't work, then you can come into the hospital, for example. Well, of course, that would not be ethical And there's a lot of discussion also here in the hospital, but at the same time, there's reality. So at the moment, I've been to a very large European geriatric conference just last week in Valencia. You know this very well. It was a wonderful city, Valencia. But it became very clear that there are so many older people that you have to make distinctions. So, for example, for every 100 geriatric patients, if the clinician is in the end going to spend much time on 10% of them only, you want to come to the 10% as fast as possible. And the reality is that people by AI are already making predictors for delirium, predicting for everything that has to do with acute admission. So I think in this age of AI, it's going to happen anyway. So therefore, I think this question is so important, because we have to foresee where to make the boundaries, because you do not want to not treat people, but you do want to get to the people that would benefit from treatment. And the same, and so this is a big debate here.
Well, all good things come to an end. Thank you much for such a fascinating and insightful presentation. Your work continues to push the boundaries of our understanding of healthy aging, and it is clear that your research will have a lasting impact on both science and society. It has been wonderful having you here with us today. And I know everyone in the audience appreciates the time, the generosity that you have taken, sharing your expertise. On a personal note, it's always a pleasure to connect with you. And I look forward to seeing you again, this time in person. So
yes, thank you very much. Thank you very much, Jose, and thank you for inviting me.